So, when we begin to look at the structure, is it necessarily unique? No. You dissect a sheep brain, any other animal, it's going to be the same. You're going to see these parts, all right? In humans, however, all right, over the geological time scale, this has been the organ of the body that has developed the most for humans, okay? Is that still the case? Not really, okay? But what we do have now, we have the ability to be learning so much about this organ. Anyone who chooses to go into anything neurology has my highest respect, all right? Because this is a complicated organ, okay? Now, this is just showing you an overview of quite a few areas and regions of the brain. We're going to find, as we move forward, we're going to see how it looks like it's got this one great big huge loop going over that corpus callosum, okay? We're going to look at how that is going to be important, and they have named it singular, okay? Gyrus. And it's going to have something that it does all to itself. We're going to see how, if we are looking at the brain, and we'll get more into this, but as we're looking at this, and if you're looking at either the left or the right hemisphere, you're going to see it looks like there's a sulci that's very prominent going across like the half on each side. That's important because each gyri, beside the one that's going to be towards the rostral, one towards the caudal, that gyri is going to be really cool. Okay, you're going to see that we have in this frontal lobe certain activities that it will carry out, where our occipital lobe, certain activities, temporal, parietal. It's amazing what this thing does, okay, that we're really not even paying attention to. As we look at parts of the brain, these ventricles that are going to exist, do you guys remember the cerebrospinal fluid? Okay, we're going to see how that gets formed, what happens to it. So, as you can see, I love this thing, okay? It is just, because every time I talk about it, I learn something new, because this thing is so amazing. Once again, if we think about the structure that's here, we're going to see that we have, once again, that gray matter and the white matter. Just like we've learned before, the white matter is where we're going to have axons, okay? Because the axons will be covered in myelin, which is that fatty protective covering, and that always gives that whitish appearance. And then when we look at the gray matter, that's where we find nerve cell bodies. Those neurosomas, okay? They're dendrites, synapses that might be present. When we look at it in reference to the brain, in reference to the brain, the gray matter forms a cortex, an outer layer. So if you think about looking at it this way, okay, when you're looking at it this way, that gray matter is to the outside, the white matter is to the inside, and the white matter lets us connect one part of the brain to another. Each area of the brain is not independent. Okay? They have to communicate. They don't have to. I mean, you know, we want them to. Okay? Under normal conditions, they do. All right? And the white matter allows that to happen because we get our tracts, T-R-A-C-Ts, that are present. So, gray matter, this outer portion, white matter, to the inside. We've already talked about how everything that we talk about has connective tissue wrappings. It's not any different here. We've already mentioned the meninges. There are the three, the dura mater, the pia mater, the arachnoid mater. We talked about them in relationship to the spinal cord, how they wrap the spinal cord. There's just a little bit of difference now when we talk about it in relationship to the brain. This time, the dura mater, the outer layer, for this organ, all right, one of the things to note, it is encased in bone, so that tells me how important this tissue is, but at the same time, bone does not directly touch this tissue. This tissue has a little bit of space so that it doesn't, that this tissue, every time we might move, walk, talk, whatever the case might be, doesn't bump against the bone. So that tough outer layer, all right, inside the cranium, that tough outer layer would have an outer portion that would be close to the bone, and then it would have a more inner portion, and it creates a space, all right? So we call that the dural sinus. Do you guys remember the definition of a sinus from part 1? Just a space, okay, that exists. This area collects blood that has circulated through the brain. That's important.
I have a question. So with the dicyclone, that's the three, the pons, the medulla oblongata, and
The diencephalon we're going to begin to associate with that area of hypothalamus.
okay. So is that white and gray matter that's in that too? Wait, let me ask that question.
No, maybe. Okay, and the reason I'm going to say it that way, okay, as we get to that region, all right, and we begin, all right, if you take a peek, all right, if you're looking at your half, okay, do you see where at this where it looks like it comes to a point? All right, does everybody kind of see where it comes to a point right there? And do you see where it looks like it forms the letter W? All right, does everybody see that? Okay, and for some, you have your pituitary gland that looks like it might be hanging off of it. Some of them are missing. All right, but I know they fell. They weren't connected. But hopefully in our purposes they are. But just saying, okay? But, and then you see the little stalk, how they got that in that white color? That's because those are axons. Okay. All right, and that W, those peaks that look like they've gotten formed in the shape of a W, we're going to find is the floor of the hypothalamus, which is part of the endocrine system. But we find it here with the brain. It's just so cool. Okay. So that's why I was like, oh, maybe. Okay. Because we're going to find that there is a connection. Okay. And we're going to have that connection to the pituitary gland. Okay. Some of it's axons. Some of it is the nerve cell bodies. Okay. So, in those wrappings, the dura mater, like I said, we have the tough outer that will be close to that actual skull, the actual bones. And then we have the portion that will begin to fold inward, give us the spaces, and we're going to also find that it will help to fold in these fissures that are existing. For if we have the two halves, okay, we've got the two halves, we've got that longitudinal fissure, it'll actually fold down into the fissure. We're going to see that for the cerebellum, because once again, two halves, okay, it'll help fold in to that area. So these have all these really interesting names, okay? Falx, tentorium, falx, okay? Foldings. All right? Tentorium is like a roof over something, okay? So these are just fancy terms to help us understand the foldings. In the arachnoid mater, which was the middle meninge, okay? Once again, we're going to see a space existing. It's termed subarachnoid. It's helping separate the arachnoid mater from the pia. And the pia was the one that was the innermost, and if you remember, can actually follow contours of the tissue. So if we look at this, the structure that's existing, we can have the pia running with all those foldings and sulci and gyri coverings and that sort of thing. When you get to dissect a brain, you can take like tweezers, or if you very carefully, like with the scissors or the probe, you can very gently kind of begin to pick at that pia mater, and it will begin to pull away from the tissues. All right? So this brain that is in the bone, very well protected, okay? So not only are we seeing it encased in bone, we're seeing it with all these meninges that are present and give us very good